In this presentation, I will be answering the following questions that involve how sugars interact with our teeth on different levels. Here are some of the sugars that we ingest: glucose, fructose, and galactose. They're all isomers of each other, meaning that they have the same atoms bonded in a different order. Galactose is also a C4 epimer of glucose, meaning the attachment at the fourth carbon differs. We also ingest a lot of sucrose, which is glucose and fructose together. When we eat food, the saliva in our mouth breaks down this food. This is done by the enzyme amylase. As you can see in the picture to the right, amylase has alpha helices and beta sheets. In the picture in the left, you can also see a calcium ion. Calcium is an essential cofactor for amylase, represented by this green molecule. Calcium binds to amylase, and this process is irreversible, meaning its delta G value is extremely negative and cannot be overcome. This is done through hydrogen bonding and metal coordination, which is a ligand binding to ions, in this case, water molecules, which then form hydrogen bonds with the amylase. These interactions stabilize the substrate binding site. Amylase also has a pH range of 5 to 10, and it's optimal at 7, which would explain why it's seen in the mouth, but not the stomach, because the stomach has a very acidic pH, and this would denature amylase. Naturally, there are a lot of bacteria that live in the mouth, so now that the simplest molecules of sugar are present, there's a source of energy for bacteria living in the teeth. There are a couple outcomes of this. As you can see on the picture on top, sugars feed bacteria. This is done through anaerobic respiration. The anaerobic bacteria are going to go through glycolysis, stopping up pyruvate and going to make lactate through the lactate dehydrogenase complex. This oxidizes NADH to NAD+ and produces lactate or lactic acid. This is going to create an acidic environment. The calcium and phosphate that make up tooth enamel have a critical pH of 5.5. So once the pH of the mouth lowers under the 5.5, the calcium and phosphate will begin to dissolve, and this is the start of a cavity and can progress to a hole. Another detrimental effect is that when sugars are broken down, they can form glycoproteins by attaching to free proteins. These are sticky and they will stick to the tooth and form what is known as plaque. One way the body provides a defense mechanism to this is salivary mucins. These are usually O-glycans, so that they're attached to serine and threonine residues, compared to N-glycans, which are attached to asparagine residues. Mucins are hydrophilic and can therefore interact with water inside of the mouth, and they have lubricating properties, and these lubricating properties protect the integrity of the tooth by having another boundary between bacteria and the tooth surface. So now let's discuss a big problem for our teeth, Streptococcus. The bacteria Streptococcus breaks down sucrose into glucans via glycotransferases. And in this specific bacteria, glycotransferases are going to catalyze the formation of biofilm, which is layering of bacteria, which creates karyogenic or cavity-inducing colonies that stick to the tooth. As you can see in this picture here, when there are no protectant molecules or mucins, the mutants can stick to the tooth enamel and come together and create this big layer of biofilm. This confirms the detrimental effect of a high sugar diet on your teeth. These are my references. Thank you.